and what we do at Xartec Salut. So Xartec Salut is a network that is led by CREP UPC, so by the Research Biomedical Engineering Research Center at Universitat Politécnica Catalunya. We are a network focused on valorization and tech transfer of health technologies. And within our network, we have 81 research groups from 23 institutions that you have on the screen. We have university, hospitals, and research centers, and we focus on accelerating these projects in the early stages of development in order that the research can reach the market and impact on the final user that usually is the patient. The areas where our researchers are focused can be divided in 7. We have researchers that do their research in robotics, blockchain, AI, IoT, big data, virtual reality, and 3D printing, among others. But what makes us relevant is that within our network, we have technologies, we have medical doctors, and we can connect them in order that the research that is made in the universities can be developed as the final user or the medical doctors would like to. This is how we work. Our work is focused mainly in 5 pillars. The first pillar that it's our basis for today. It's providing the community with training to PhD researchers, future spin-off founders that may be interested in entrepreneurship and creating a company based on the research. We create in Xartec Salut, we are a space for idea validation, as I mentioned, between hospital and technologies. And we develop or we have spaces for co-creation. We have created several activities for this and the HealthTech 2030 that I will mention afterwards, and also the Xartec Salut Share and Connect. And for the last 2 pillars, valorization and tech transfer, this is directly supporting our research groups, the research groups from the network, and we provide direct support to them. What I would like to emphasize is that this session is the last session of the Entrepreneurship Academy. This series of 6 experts sharing their experience in very concrete areas. So I invite you to have a look to our YouTube channel. You have the rest of the sessions there, and this session will be also in our YouTube channel. If you want to have a look afterwards, or if you want to share it with your partners. For the session, what I would like also to share with you is that as in the rest of the sessions, I invite you to participate on the HealthTech 2030. This year will be focused on talent, and it will be the 3rd edition of our main event, and will take place on the October 10th in Casa Rius here in Barcelona. So I invite you to have a look in that and subscribe to the event. You have the webpage here, but you have also the QR. The same for the hackathon. The weekend before the HealthTech 2030, we will have a hackathon where we have committed industry partners that can provide challenge to you, and you can form teams with multidisciplinary profiles such as medical doctors, engineers, PhD, postdocs, so you can together solve the problem that they have. So as in the case before, I invite you to participate on our event. All this information it's on our webpage, either on the HealthTech 2030 that is specific for our innovation challenge and the event, or either at Xartec Salut if you want to know more about us. This is also our social media. We are very active on LinkedIn and Twitter, so have a look on that. If you want to contact us, you have also our email here below. Yeah, I will very briefly introduce Marie-Eve, but she will explain later her profile. Marie-Eve is a pharmacologist by training. She did a PhD, and after 2 postdocs, we were talking before, one on the industry and one on bio, Valdebrón Institute of Oncology, together with Laura Solchek. They founded Peptomyc, and she's currently holding the position of Chief Scientific Officer at Peptomyc. Thank you so much, Maria, for coming today. I will pass the floor to you, and you can start presenting the screen. Thank you so much.
Thank you so much, Alberto. Let me try to share my screen. Here we go. Okay. You can see it?
Yeah, perfectly.
Great. So I'm not putting it in full screen right now, but we can do that later, because I've included some Mentimeter activities to try to have really a conversation with you today. But first of all, thank you so much, Alberto and Cristina and Xartec, for inviting me today to share my experience with you, from research, academic research, to transition into biotech and delivering value to society. And I will try to stick as much as I can to this, or to use this story to really explore this topic of how to make our research reach society and impact the lives of people. But I would really like to have this session as useful for you as possible. And I know that online conferences like this can be challenging to have you engage and so on. So I hope that with the Mentimeter we can already have a first conversation, but I invite you to interrupt me at any point in time. If you have questions, you can raise your hands, you can write them in the chat. Alberto and Cristina will let me know. I will not read the chat, but they already will help me look into the questions, and yeah. This is meant to be a conversation as much as possible. And yes, let's start. So as Alberto was saying, I'm co-founder and chief scientific officer of Peptomyc. We are a biotech company. We are a spin-off from the Valdebrón Institute of Oncology, which is a research center that is really translational, linked to the hospital of Valdebrón, so really close to the cancer patients. And we are developing new therapies for cancer treatment. And what is special about Peptomyc is that these therapies, they target MYC, which I will show you a bit later, is an oncogene that is deregulated in the majority of human cancers, and that has been undruggable. There's no MYC inhibitor approved for treatment of cancer in the clinic yet. So we're the first one to do this. In this sense, we are pioneers. And as you can imagine, that also brought some challenges in communication with different actors and trying to share why we think it's important and why we think that it can make a difference in the life of patients and of these people. So first, I would like to, the key topics I want to cover today or to touch on through this story is these topics that have been announced on LinkedIn, making science and research accessible, engaging with the public, building impactful relationship and beneficial collaborations. And you will see that I'm trying to include in this all the actors of the value chain, all the stakeholders, all the people that have different contribution or that can have different contribution to the success of a project like ours. So we've created Peptomyc 10 years ago, in December 2014. So there's a lot of people and there's a lot of experience through this. I will not be able to cover everything. I tried to make a storyline that has more or less a thread that we can all follow. But feel welcome to ask questions about any of the actors of the sector. We've interacted with many of them so far. And so before jumping right into the topic, I would like to use this tool, the Mentimeter, to get a bit of your pulse on a very simple question. That is, what words come to your mind when you think about biotech company? So I will put maybe in the chat the link. You can use the Q code, or I can try with the chat to share the link so that you can access the webpage, and you can answer directly this survey on the website. And I would like to give you maybe a couple of seconds or minutes to start answering this first question. And I have a second question for you also after, but let's see if this works. Right. Okay, we're starting to get some answers. Patients, medicine, corporate, that's great. Roy, I guess it's return on investment. Biopharmaceutical, antibodies, yeah. Medicine, antibodies come often, proteins, bacteria, biology, biopharmaceutical, illness, corporate research, that's great. Mm-hmm. All right. So you can keep adding answers, as many as you want. I think that from this, biohacking, that's great also. Long to develop. Yes, it is long to develop a new drug. It takes long to develop a new drug. That's great. So we can see from this word cloud that you are a really focused public, that you have a really, we know who is assisting to the seminar. Like the seminar was directed to master student, PhD, postdocs, young investigators, and so on. And this is reflected in the words you're choosing, in your understanding of all this entails into biotech. Still reading this. What else did we get? IP protection. Yeah. Knowledge, knowledgement, mitochondria. Oh, that's great. Hard work. Yes. So we can see the focus that you guys have when you think about a biotech company. And this is great. I want to share you now. A couple of months ago, I gave a class to a group of, it was business design. So a group of people that have backgrounds in arts, in mod, fashion, in different industries that have nothing to do with what we are doing in health, or that have, so more representative perhaps of general public with little background in biotech. And here are some of the answers that they share. So we can see that there is some overlap. Regulation, biochemistry, challenging, pharmacy, medicine is there, testing, research. But there were also concepts that they had in mind, which I thought was interesting to raise. For instance, anti-capitalist was something that one of the students raised. As I asked her to clarify this aspect, she basically said that she had in mind this idea that perhaps there's this alternative view of our reality, that pharma is not trying so much to cure people, but there is this huge market opportunity for people that are ill to sell drug to, and that biotechs are a bit the Robin Hood in this, that are trying to, develop new drugs and really solve the problems, while big pharma is not always doing that in our perception. There was another one that was, the myth of relief. So curing health or prevention versus having drugs that solve things quickly, restrictions, regulations. And with that, I just want to illustrate that also in the general public, we are aware, but there are different perceptions on what we are doing when we are doing drug development. And that has to be part of our thinking when we communicate about science. We'll come back to this, but there are myths, there are preconceived ideas, there's language challenges that are part of these communication aspects that we need to take care of. And actually that brings me to a second question, which I would like to ask you right away, is what, in your opinion, are the main challenges in communicating your scientific advances to the general public? So there again, maybe I share the Chrome tab here, the Mentimeter, and I can go to the next one here. And if you want, you can answer, short answer, but what do you see as the main challenge when you want to communicate your own science in general?
Can you share it on the chat, Maria?
Yes, it's the same link, I think, but let me check.
TSN mismo link.
Sorry for that. So
Yeah.
it's the same link. Okay, sorry.
Yeah. Are you able to access it?
Yeah.
Great. So, and then I will give you again, maybe 1 minute to try to answer at least one answer and start gathering some answers. But we will come back to this towards the end of the session. Yeah, complex terminology. Yeah. If I can share again the link in the chat. Thank you, Christina.
Thank you so much.
And then maybe I can do this. I don't know if I can. In Mentimeter, close this, put it full screen. Yeah. So yeah, some of the challenges that we are aware of, complex terminology. If we try to convey into simple terms complex science, that's one challenge. Knowledge gap. Synthesis, data overload. Communication challenge for the target population. Tools and time. So if you're not too shy, is the person that answered tool, would you like to expand a bit on this aspect? What did you refer to? I don't see you. I can only hear you.
The person who wrote that? Yeah, it's really shy, I think.
Okay. New areas of science. Maybe tools refer to communication channels, difficulty of sharing complex processes with people with no knowledge, limitation to access public. Yeah. That's great. So you can continue if you want to enter some terms, and as we advance, you can add more of them. I will leave the Mentimeter open, and we can come back to it later in the presentation if you like, to see what is the final result. So, but these are really key challenges that are the obvious one, how to make our message clear and how to make it really reach the people that we are trying to reach. So, okay, let me tell you about Peptomyc a little bit now. So the problem we are tackling is, of course, that there are still too many patients that die of cancer. And the current therapies, I mean, they have improved tremendously in the past decades, but current therapies often target redundant functions of cancer cells, which leads to resistance to therapy and tumor recurrence. Also, several of these drugs are still toxic, so they reduce quality of life of patients. And as a result, I've put here the total cost of cancer care in Europe, but in the world, it's very similar. I mean, that's huge cost to society, and that's why it's important to find new and better ways to treat cancer, of course. So when we think of cancer resistance to therapy, why do cancer cells develop resistance? Often the drugs target cell surface receptors or enzymes that are more accessible, that are more easy to drug, but that also happen to be functions that are redundant in cells, meaning that if we block one of them, the cancer cells often finds a way to compensate for the block function and to thrive, to find a way to activate a compensating mechanism, and evolving. So it's a bit like the guacamole. We just hit one mole, but there's another one popping up immediately. And so our approach is to find instead common conduits that cannot be compensated by the cancer cells. So what if we could find common functions that are not redundant at all? And one of these functions is actually MYC. Now I can put maybe the slide as, let's see, as full screen. Now I really don't see you at all. So our solution is to target MYC, and MYC is one of these non-redundant and essential cancer enzymes. So MYC, it's a transcription factor. It's been identified in 1978 as a part of a virus that was causing cancers in chicken. And so in normal, it's been studied for the past 45 years. And in normal cells, MYC is actually a transcription factor that coordinates cellular programs, several of them: metabolism, cell division, so cell cycle progression, cell division, survival, and many, many different things, even the immune system. So what happens is that in normal tissues, MYC is usually present at very low level or even absent. But for instance, if we have a wound, if we get hurt, the skin cells will need to proliferate to close the wound. And then MYC gets transiently activated. So for a very short period of time, MYC is activated. Once it is activated, it's like this orchestra director. It coordinates the metabolism, the cell cycle progression, all these different programs so that the cells proliferate, and then MYC disappears. So it has a very short half-life. And this is what happens in a normal context. However, in cancer, what we find is that MYC gets deregulated, either because there's gene amplification, so the gene of MYC gets amplified, there are many copies of MYC, the levels of MYC are really high, or because MYC is sustained, so its degradation is impaired and it's always present in the cells. Because of that, it always drives this metabolism, cell cycle progression, and cell division, so they are driven in an uncontrolled manner, and this is what results in cancer. So because of its essential role in cancer processes, MYC really has become a most wanted but undruggable cancer target. As I was telling you before, there's no direct MYC inhibitor that has reached clinical approval yet. So why is that? There are two main challenges. The first one is that MYC is what we call an intrinsically disordered protein. It's like a spaghetti that changes shape all the time. So normally when we want to design drugs for specific targets, we use the analogy of a key in the lock. And instead, it's like we're trying to design a key that would bind a spaghetti. It's really hard to have specific interaction, the specific key for the specific lock in this case. In order to achieve that, we need much larger molecules that can have a larger interaction surface, so that can bind MYC with specificity. The other challenge coupled to that is that MYC is a transcription factor that is located in the nuclei of cells. So these big molecules that are specific for MYC, they have a hard time reaching the nuclei. It's not easy. In order to function, MYC has to form pairs with MAX. So this is what we are seeing here. We see this dimer, the pair MYC and MAX, that together bind to the DNA target gene and activate these processes: proliferation, transformation, even apoptosis. This happens through this region here that is called BHLHLZ. It's just a small portion of MYC, but it's a portion of MYC that adopts a certain structure when it's bound to MAX and bound to DNA. So when I did my thesis some time ago, I was studying this structure. So Alberto was mentioning my background before. I've trained in pharmacology all my university lifetime. I've always been interested in pharmacology, in drug design, in understanding actually how does the structure of protein and how the structure of peptides and hormones and small molecules regulate our health and our disease. So I've done molecular modeling, I've done some cell studies and binding studies during my master. During my thesis, I really focused on these domains of MYC and MAX. I was doing a lot of purification of protein, mutation of these domains, and the goal there was to understand how does MYC bind to MAX, how does it bind to DNA, and could we use this structural information to design better drugs? That was basically the topic of my thesis. And then I did a first postdoc in industry in a company based in Montreal that is called Technology, where I was really immersed in the world of pharmaceutical development. I learned for the first time all this value chain, how is a peptide drug developed until market approval. So that was really a fantastic and eye-opening experience. While I was doing this postdoc, I got a call from my PhD supervisor that invited me to see the talk of Dr. Laura Saucek. He had invited her to the university where I had done my thesis. And I was really excited to meet her because I actually knew Laura on paper, because Laura had published an article while I was doing my thesis that showed a first translational approach to actually apply the structural knowledge to the design of MYC inhibitors. So what had Laura done? She had taken this domain of MYC, this dimerization and DNA binding domain of MYC, and she had introduced 4 mutations here to imitate a bit the structure of MAX. And by doing that, she created this molecule that she called HomoMYC, which is able to form homodimer pairs. This is something that MYC cannot do itself. So HomoMYC was able to do that and to bind to DNA, and it was also able to bind to MYC and to MAX. But what was really surprising is that when Laura expressed this HomoMYC transgene into a transgenic mouse model, HomoMYC was able to block MYC function without causing dramatic side effects. And that was really an important aspect, because for a very long time, people were just scared of blocking MYC because of its importance for normal tissue proliferation, like intestinal renewal and wound healing and these kind of processes. So the fear was that if we block MYC, we would have side effects that are similar to chemotherapy, for instance. So instead, Laura produced these transgenic mice that expressed the gene coding for HomoMYC. And what she showed was that in mouse models that had lung tumors, lung adenocarcinoma, HomoMYC was not only able to be expressed safely, but it could cause eradication of the tumor. And that was really a game changer. It showed for the first time that we could block MYC safely. That had never been done before. So she published that in Nature, later on in Genes and Dev, with more aggressive models and more characterization. And that was really the first opening of this possibility of blocking MYC in vivo for cancer cure. Still, there was really a big challenge into turning HomoMYC into a drug. Actually, the biggest challenge was that HomoMYC is a big molecule. It's at least 40 times the size of normal drugs. So people were writing in the literature that HomoMYC is too big and too bulky to be delivered into the cells, and that it was essentially just a proof of concept and would only work as gene therapy, which unfortunately for cancer treatment is still quite challenging to do. And so that's when Laura and I met. She had published this article. I met her at the presentation, and after the presentation, I got to spend time with her, and we started thinking about this potential project of what if I try to purify HomoMYC for the first time, since I know how to purify this BHLH-ZIP molecule. And what if I start to play with it, maybe changing it, altering it a bit to render it into a potential drug? What if it could become a drug? How could we turn it into a drug? And so she considered that my CV was good enough to apply for a postdoc. I started writing tons of grant application, fellowship application, and eventually we got some funding to move from Quebec to Barcelona. So I joined her lab initially in 2013. She was just starting her lab. She had started 1 year before. So in terms of protein purification, there was nothing. At VIO, there was nobody else doing this. So for me, it was really an opportunity to build the protein platform of my dream. Whatever I was missing during my thesis, I could implement there. It was a new experience for me also in terms of working with mice. I had never done that, and to make new friends at work. And these happens to be friends that also enjoyed working late at night, so it was a great experience. And one of these late nights, I managed to purify omomic. I actually labeled it with a fluorescent molecule and treated some cells. And after 15 minutes, started looking in the microscope, and I saw that omomic was able to actually penetrate the tumor cells. So that was extremely exciting. It was a spontaneous property. It was a surprising property. We were not expecting that omomic could be a cell-penetrating protein, but it did display this property. So that opened the possibility of using it eventually as a drug directly as omomic. And so that also opened the opportunity of patenting omomic. So here comes the first aspect related to communication and dissemination and patentability that I would like to discuss with you. When we talk about making an innovation patentable, what's important, and maybe you've seen that in the past few sessions, is to know if our invention is novel, if it also has this inventive step. And how is that determined? It's often determined with state-of-the-art search, which asks the question, could somebody skilled in the art have come to the same conclusion? So it has to be non-obvious. And as someone else claimed the same innovation. That's another search that is performed when we try to apply for a patent. Later on, there's also a freedom to operate search. If the patent is applied, is there any other patent that could prevent you from exploiting this innovation commercially? We also learned through the years that there are different patent laws in different areas of the world. So one aspect is that for European patents, first of all, you need to have much more proof before applying for a patent in Europe versus the US. In US, you can patent an idea. In Europe, you need to have data supporting that idea. And in the EU, actually, the inventor loses all potential patent rights if there is public disclosure of the innovation before applying for a patent. Whereas in the US, there is 1 year grace period for public disclosure. So here comes my third question for you. In your opinion, do you think that a poster or an oral communication at a conference constitute a public disclosure that could prevent the patentability of an invention? So I will go back to the Mentimeter and ask you again for your input if that's something acceptable to you. Let's see if we go to the next question. So we'll give a couple of seconds more. So that's interesting. We have a lot of yeses. 8 yes, 2 no. Can give you a couple of more seconds just in case. All right, let's go back to the presentation. So actually, we checked with our patent lawyers this question, which is really important to us. Okay, now you can see it. And actually, presentation at conferences by themselves, they are not considered public disclosure, which in our case helped us with the later patent application. So we had disclosed the innovation in an abstract, which got published, and actually conferences are considered private, invitation-only events. However, what's important to know is that in the US, a random person on the street bearing a sign that just talks about your idea or disclosing your invention is public disclosure, and that is sufficient to prevent you from patenting in the US. So you can disclose things at conference, but be ready, because if somebody else discloses publicly or on Twitter or about your presentation, then it's really public, and that really prevents you from patenting. Okay. So we had created this first patent application. We could create Peptomyc. Peptomyc, we've created it at a specific moment in time, driven by money needs. we created it to apply for grants, because this tech transfer and drug development, you've mentioned it in the first case, it's really expensive. You start spending money on patent lawyers, you start spending money on so many different things, advisors. And so we created Peptomyc with the idea of applying for grants and the timing in mind for that. So the founders are Laura and myself, the institution, VO and ICREA. And the goal of Peptomyc is to develop our drugs until clinical proof of concept. So phase 1-2 clinical trial, where once there is enough data, we can license them to big pharma who have the capacity for completing the commercial development and reaching the market authorization for the drug. So that's the business plan of our company. And this is just in a nutshell what our innovation is. It's basically using cell-penetrating homomic mini proteins that have this capacity to bind to MYC, to bind to DNA and occupy the MYC binding sites as homodimers or as heterodimers with MAX, all of the which block MYC activity and selectively kill cancer cells and not normal cells. So the hypothesis is that will lead to increased patient survival and less toxicity compared to current drugs. So when we created the company, the whole stakeholder chain, the whole value chain that we have to deal with and to whom we have to communicate involves many different actors. Of course, there's the team. So we need to be able to build the team. We were 2 employees, Laura and myself. Eventually, with some funding, we were able to be 4, and now we are 16 employees at Peptomyc. So with all these employees, there's actually already an effort of communication, but there's also all the extended team. We have a lot of advisors. We have scientific advisory board. We have key opinion leaders. We have business advisors to whom we need to be able to communicate the essence of our science and to engage also in the project. There's a lot of partners because we have to perform experiments. Some of them we can perform in-house, but a lot of them we outsource, like most virtual biotech companies. So there's also communication effort with these different partners, scientific partners. In this case, I will give you some more example, but it's really important also to position our project for them, to make this partnership a success and to have the right priority level for them. There's the patent attorneys, there's all the agents regarding fundraising. We communicate when we write these grants, we communicate when we go to see venture capital, and so on. We had to communicate with university for the last license, all the operations that enable us to expand the project, and eventually communicating for the clinical trial execution with the hospitals, even the public communication that ultimately also reach the patients. Communication with partnerships, there's a lot of other companies we have to partner with to develop the project. So there's a lot of different aspects into this. Perhaps the first tool to communicate is to define this business plan. We wrote the first one in 2014. It's about starting to think and define really clearly what is our company mission. What value do we bring to the world? What do we want to make as a difference? What's our differentiator versus the competition? So what makes us unique? It's really an important aspect to communicate in the business plan, but in general, in all these other stakeholders' communication. Then in the business plan, we had to define timeline, budget, and so on. And our first business plan was wrong in so many ways. I mean, we made projections that were different every 2 years and so, but it's at least a plan, and you can at least advance with the plan and help define a bit more who you are as a company and what you're trying to achieve versus what everybody else is doing. One really, really key aspect for the success of a project like ours is having the experts' advice and having the right network. In that sense, we were extremely lucky because being part of the Valdebron Institute of Oncology means we were surrounded by oncologists and key opinion leaders in the cancer indications we are working on. We also are part of networks, European networks, and so a lot of interactions with principal investigators and experts enable us to better articulate what makes us different, and also having them engage makes them some sponsor of our work in a way. So it was really important, and it's really something we've done from the beginning, is to really nurture this relationship, learning from these people and having an open conversation about what we have in mind. Is it feasible? Is this indication better? What would be the selection criteria that we need if we want to go in this type of cancer? Asking advice on many different aspects. What will be the patient's perspective, and so on. One example I can give you is at the beginning of the project, I was producing the protein in the lab myself, treating mice, and I was only having access to very small quantities of the drug. So I was using intranasal administration to treat the mice. Asking advice to pharma companies and to these oncologists, they told me that yes, it's feasible to develop a product that is administered intranasally, but it's not very convenient for the patients, and it's not very convenient for the regulatory agencies because it's hard to dose. It only goes into the lung. You would not treat all the types of cancers. So based on this feedback that we got from them, we changed from intranasal administration to intravenous administration. So we really pushed our efforts and our limited resources to make it feasible to reach intravenous administration. So it's just to give you this perspective, perhaps, that this wanting to engage the network, the right people, it's also a two-way conversation. You get them engaged when you ask for advice very often. So that's one tip here. We wrote tons of grant application. I've said that already. Here, the success is really...
Sorry, Marie-Eve, I wanted to ask you, because I think this is something really important on this sense. So in the terms of you're talking about a drug, but you changed the way of delivering because the feedback you had. So I think that's something really important for the community also, because we have had, for example, Alphonse Carnicero, you may know him, and he was saying the same. So it's like making small prototypes in pharma is very different, but like making prototypes and asking the end user which is the best way, no, to do so. So I think this is something key aspect. But I wanted to ask you also, because of your background, you talked about the first business plan. I'm sure you were helped by a consulting company or business experts on the development of that. But you were saying I was producing proteins in the lab, but how that switch to the business part was for you? Because you are talking now as the Marie-Eve from 2024, but the Marie-Eve from the past, how that was?
Yeah, that was really interesting. I mean, I think that when I came to Barcelona to work on this project, I was ready to do anything for this project. That was the first premise. I was not focused on having an academic career, never in my life. I had done a first postdoc in industry because I wanted to explore, So I was really coming with an open mind of, let's see what happens. So for me, when came the opportunity of creating a company, it was really exciting, and I was all in this. What is funny and at the same time sad from this industry background is that when I trained at Terra Technology, I stayed with them for 4 years. Okay, the first year and a half, 2 years, I got to know, I don't know, 50 partners, team workers, and eventually there was a layoff round, because there's often cycles in the pharma industry. So there was a layoff round, and these friends that I had made, they got disseminated throughout the industry everywhere. But they were people that were really good in pharmaceutical development. So CMC, regulatory, logistics. So through them, I got to know a little bit of everything without knowing much about everything. No? But I think that it gave me some sort of confidence that I could learn, because I could call them. That was the first thing. And then, as you say, we had also consulting. We were working with Antares Consulting, Giuseppe Luis Falco, that you know very well. We had a couple of consultants that gave us also their business perspective. And that was also important, to have everybody learning the same language or discussing the same aspect. So I think that from my side, it was just, I wanted to jump or dive into the topic, whatever it was. I was okay with doing science and doing business. And I think this is what makes my job as CSO still really exciting, because I get to do both. As a co-founder and CSO, I got to do a lot of business meetings, a lot of science, and I think I never get bored in this. But I think that to me, it's just that. It's just wanting to learn so much and finding friends to help you along the way. It's I don't know if I'm answering your question, but that's...
Yeah, for sure. For sure, I was thinking. No, for sure, you answered it. And that's something important, no? Because this open mind that you had, I think this is the key point, no? You had a very open mind when you started, when you thought that maybe this project could reach the end user, so you were open to that. So I think the important message here is that to have an open mind to new things, because business is something that is some kind of different from science, but it can be learned, because scientists have a very strong background. This is something that can be learned for sure.
Yeah, absolutely. It's much easier to learn business when you're a scientist than the other way around. That's for sure.
Yeah,
for sure. But perhaps the other message that I really want to stress here is that when we went to ask for help to these medical oncologists or to these people, they respond to, like, the expert. The people in industry, they want patients to get better. So when they see some project that has potential, they're interested in making it happen. So that's the other message maybe I would like to tell is, at the beginning, I was a bit shy of asking for help to these key opinion leader and great people. But with the experience, what I learned is that it's the best thing that you can do, because they want to help the project process proceed. So very often they are becoming allies, and they are becoming also spokesperson for you. They also talk about the project to different people, which impacts positively this dissemination and this communication that you want to achieve. Yeah. Okay.
Thank you for the feedback.
No, thank you for the question. Regarding public grants and loan, the message here is we've applied to a lot of those. I've said it several times. And I say it because it's important, because every time you get a rejection, doesn't mean the project is not good. It means that there's something that has not been communicated in the way that the evaluators were receptive to. And it's the same thing when you try to communicate with different audiences. It's really trying to get to know what does the other person wants to know, what is important for the other person, and trying to target that message as well. So every time we rewrote and reapplied for grants, it's just an exercise in clarifying things, in clarifying some aspects, and really reading the feedback and listening to the feedback and rewriting thousands of times if needed, but it helps clarifying the message. And that's an important training tool, I would say. So for me, writing grant application, it's a training tool. It's something that should be seen as this way. Then when you get it, you can celebrate. That's great. The other thing is, it comes back to a bit the feedback with the pharma. This is a slide that I gently asked and got authorized by Dr. Luis Pareras, who is an investor at In Vivo Venture. He's a managing partner and founder at In Vivo Venture, Barcelona-based investing company specialized in biotech. And basically, what this slide shows is what does VC value in a first meeting. And these first meetings are really important. It's where you actually can open the door to funding to your project. And these VC will eventually, the venture capital that eventually invest in your project, they will become your supporter and they will also become your spokesperson. So what do they value the first time that you go see a VC? What do they look for? One of the first aspects is the team. So is the team strong? And you can see here it's already part of this first aspect, the communication skills of the CEO or the founders teams. People that join this meeting, you need to develop a clear message that you can communicate in the very first meeting, and that's something that is valued. There is vision, skin in the game. So the vision you define through business plan writing, through this grant writing. The second aspect is, does it solve a big problem? What's the market potential? What are the possible extension and so on? And then the third aspect is the proof of concept. So all the sound and strong science, of course. But the ability to communicate, of course, it's really important for these first meetings. So some of the activities that we've done during these times, I'm calling this normal times by opposition to something that comes later. But this is when we did not have a high impact publication yet. This is a time where we are generating a lot of data, a lot of science, but we don't have, again, a strong result to disseminate or to announce. What do we do during this time for communication and dissemination? So one thing we did with Laura was fundraising for Worldwide Cancer Research. Worldwide Cancer Research is one of the first funding agencies that supported our project with a grant. So they give us funding for 3 years. And we did this project. We went to hike Mount Toubkal in Morocco, and we fundraised for this agency. So that was like social media dissemination. It was a way also to meet other people, volunteers that were doing fundraising also for the cause, for cancer research. And it was one way that we could communicate our message through social media, through meetings with these people. And so that's one way to reach general public. Also, Worldwide Cancer Research asked me to write a small article about a day in the lab. So what does it look like? So writing on a web page from the funding agencies, whenever these opportunities arose, we took them. It's some time to invest, but it's really good dissemination for our project again. There are awareness campaigns. In Spain, we have several of them. One of them was Con los imprescindibles, with the indispensable researchers. So taking nice pictures and showing that who are the scientists. That's one way to do public dissemination on Facebook, on Instagram, so general channels. And another thing with this was sport events. So there's now a club at VU that is called Running for Research, and we participated into some races to fundraise and trying to disseminate for fundraising for cancer research. So that's kind of an indirect opportunity to talk about our science to people at the event, before the event, and after as well. And for these, I mean, the communication tips, it's really, we have to tailor the communication level to the public that we are, of course, dealing with. So you've mentioned already in the first questions, the challenge we have. One of the challenges is that there's a sort of language barrier. So we need to adapt the language. We need to be to the point. It's good if we can practice before a bit the key message. What is the most important message I want to transmit? Adapting the language level. And to me, it's also really important asking questions and asking for advice, as I mentioned before. It engages people. It helps you understand their level of understanding as well. And then try again. It's an iterative process and it's experience. So take everything as a learning opportunity.
Well, where do you seek for advice for this kind of communication, general communications?
Sorry?
Where? I mean, if I was in your situation, imagine I am a PhD doing my research. Where did you find this advice? Did you ask, for example, I can imagine that you asked your family members. Do you understand what I'm saying? This
Yes.
is normally what everybody does, no?
That's
But
a good.
anything professional that could be helpful, or within your institution?
I mean, for us, it was really working with the Valdebron Institute of Oncology, with the communication department there. We see Amanda here, Amanda Wren. She's been helping us a lot with making the message impactful. So sometimes it's simplifying, or sometimes it's finding these images, these examples, and trying to take note of these ideas as they come. But asking the communication department of the research institute for help. As you say, asking family, it's also another good reference. It really depends the public you're trying to target. Testing things with your relatives, testing messages. I think that's a good advice. Yeah. Okay. Eventually, after applying to all these grants and seeing all these venture capitalists and trying to communicate our message to all the different stakeholders, some fruits started coming out. We started receiving some prizes for our business plan. We started receiving some prizes for the Premios Emprendedor, grants from the ministry as well. So that gave us a bit of money to actually advance in developing the drug itself. We also closed 2 rounds of investment. The first one in 2016, that was with business angels and with first investor, which is now in Vivo, so Health Equity and Luis Pareras. And the second round with Alta Life Sciences with Montse Vendrell. So together, these 2 rounds, they gave us a lot of gasoline to advance everything that is called regulatory preclinical until reaching clinical trials. One of the key milestones that we needed for this project was to have a manufacturing process in place. It's a challenge because we're not doing an antibody, we're not doing a small molecule, we're doing a new type of protein that nobody knows how to produce. So we hired Virginia. I hired her in 2017. And we started working together. Virginia is a biophysicist. She knows protein production. We started working together. And both of us had worked in production in our lab. You can see in this nice picture, the first time we purchased a big column for chromatography that was almost as tall as her. It was really exciting for us. We're going to produce a lot of protein in the lab. But actually what we needed to do was to transfer this to a CDMO, to a GMP certified lab that can produce material that can be injected to patients, right? And that's been really a communication learning experience. I'm showing you this image. I don't know if you're familiar with the Hitchhiker's Guide to the Galaxy. It's a bit of a geeky image. They have this Babel fish, which is a fish that you put in your ear and that translates everything spontaneously. So when you speak to somebody with a different language, you understand everything that they have. And I'm putting this here because this is what I wish I had at the moment, to learn the language of somebody that works in GMP with large-scale tanks, with fermenters of thousands of liters, with reduced risk for production, with 8 hours shift, little flexibility, whereas my way of producing it and Virginia's way of producing our protein was small lab scale experiments. I need to come on a Saturday, that's fine. I can come in the evening to centrifuge. It was a different thing. It was a different thing completely. We knew omomic biochemically better than anybody in the world, that is for sure. But the CDMO we needed to work with, they don't know omomic, but they know their upscale process for other drug. They know their fermenter, they know their tank, they know the processing time it requires. So when we started imagining production at large scale, was really a big effort in communication to align these two visions together, right? So I would say that in this sense, it was really like team building. There's this graph, I don't know if you've seen it before, but it's about how a team is effective over time. So when you start beginning team working, you form the team. And this is where you start learning to work together, establishing dynamics, starting to understand where your limitations are in mutual understanding. And then you start seeing where the language barriers are, because when there are misunderstanding or there are things that you cannot resolve easily, there's this storming phase where you actually brainstorm and try to troubleshoot the communication, and eventually you establish some norms and the team starts performing. So in our effectiveness to transfer the process to the CDMO, there was also this team building period of time, evolution. But there is more than that also, because when you transfer to a CDMO that doesn't know your company, it's critical to be able to communicate the value of your company and your vision very, very clearly, because that determines actually the quality of the people they put on your team. Who is going to work on your project? Do they put the highly experienced people, or do they put the new people that begin? So with this image here, what I want to show you is it's really important to define really early on what makes your project special. What's your vision? Who will you become? You know, in our case, we're targeting MIC. MIC is important for many types of cancer. We can help many, many different cancer patients type. If this works, if you help us make it happen, you can win a lot through working with us. So today we are tiny, we look like nothing. I come to you, I don't have the experience to even understand everything you're telling me, but we're going to get there. Believe me, we're going to get there. So that's kind of a message that I think is super important to define. How you position yourself, how you are special, how your project is special, what makes you different from all the other ones. And that is how you manage to get, again, engagement and get valuable input from their side, and have them engaging competent people to work with you on the project. Okay. Now I would like to talk to you about 2019. I still have a couple of minutes. Yeah. I'll go to this and then maybe I skip a couple of slides after. 2019 was really a big year in terms of communication. So we were starting to collect some prizes. We got some prizes for business development, and things were going well. And I had been writing grant application for a grant called SM Instrument from the Horizon 2020 program for almost 2 years. They had this thing that they call Seal of Excellence, which is, we tell you that your project is excellent, but we don't fund you yet. We don't give you the money. So I had collected 6 of them. It was really hard work. And so eventually, we managed to publish the results showing the first proof of concept that omomic could be used as a drug. And that was done in vitro and in vivo, and we published it in an open access journal in Science Translational Medicine. That year, through VO, we got a lot of press appearances. So again, the communication department did a wonderful job because they really nurtured the communication with the journalists. We appeared on TV, we appeared in radio, in RACU, we appeared in newspapers, name it. We appeared everywhere. We even were solicited or asked by Nike to participate to their campaign, which was for the new T-shirt that they had. So the campaign was talent has no shape. Talent takes multiple shapes. And so this is us on the street on Passage de Gracia. I mean, our picture was huge. It was 1.5 floors high, next to Piqué and next to Messi. And actually they had organized this very nice photo session with Messi and with Piqué at Camp Nou. And the photo session happened to be just the same day as the interviews for the EIC grant that we were applying for. So there was 2 stage, writing and then interview stage. And of course, the interview in person in Brussels was the same day that we were invited for the photo shoot with Messi. So we had to decide between both. And what do you think we decided to do? Well, we did the right thing. We went to the interview, even if we were not sure we would get the grant. And eventually we got it. So we did not the communication activity that day, but the grant activity that day. And Nike was really nice because they enabled us to have the photo shoot 1 week later, but we missed Messi and Piqué. So this photo, I mean, it was diffused everywhere in Barcelona. We were in all the metro station, in all I had friends sending me pictures from bus station, everywhere. So that also made a lot of people talk about us in addition to all this press release. I think that the message here is, again, the communication department from the research center, they did a wonderful work. So to me, that's a key good relationship to nurture. And it's also about how you publish your research. We published high impact, and they really helped us making it high impact worldwide. So that was also key. Okay, then there was COVID. That was really complicated. There was, with COVID, no face-to-face meetings, material shortage, increase in cost, everything. We were facing value of debt right before starting the clinical trial. I mean, the COVIDs really arrived just before the clinical trial for us. And so I would like to reiterate here again, these are all the partners most of the partners we were working with, how important it was for us to communicate why we do things, why it's important what we do, how we want to achieve it, what it is that we are doing in the clearest way possible with each of these partners. So it's not just when we want to disseminate and communicate, it's not just doing it with the press or but engaging so many people, because these people talked about us as well. These people helped us advance the project. And this is what makes you advance and reach the milestones that enable you to communicate eventually. So really being focused on executing, advancing the project, and engaging these people by defining very well why you do things. We got another investment round even during COVID, this time with an industrial investor, so an investor that is linked with pharma. This enabled us to really consolidate the team. We could hire a chief medical officer, chief financial officer, a chairman for the board. And that brings me to talk about the team. So we hired a lot of different level of people during this time. And for me, I would say that a key aspect also in communication is when we select people that we trust, that will bring value to the organization. It's critical to groom our talents, to help them thrive, to ensure talent retention. In our case, we have project managers that have actually done their thesis with us, 7 years ago. They know the project better than anybody else. We like working with them. By nurturing them and making them grow in their roles and evolve and making them happy, they are also becoming spokesperson for us. So this is a nice picture with the team. It was taken a year and a half ago already, but these people, they have their own network too. They disseminate, they have family, they have relatives, they talk about your project as well. And so they contribute to also this dissemination effort, of course. So I think that another message is it happens at all levels, this communication aspect. Okay, we don't have a lot of time. I just want to tell you that now we have completed the clinical trial, the first clinical trial. In advanced solid tumor patients, we've confirmed clinical safety and clinical activity in monotherapy in our first in-human trial. And we've seen there 49% tumor reduction and 6 months stabilization of the disease in late-stage pancreatic ductal adenocarcinoma patients. So these are the really first encouraging signs. We are super happy about these first trial results. And we've published them last February in Nature Medicine. And this, again, is an open access journal. So that's another aspect that is important, is making our research accessible by publishing in open access journal as much as possible. Because this gives the public the access, the full access to the full data set, not just the summary by the journalist, which is quite different sometimes from the study that we are working on. When we've published that, it has also had a lot of impact in the news, and both times in 2019 and this year, we have a lot of families and patients reaching out to us. So that's a personal experience as well, but I've had patients reaching out to me directly on Facebook, writing emails. I mean, our email address are there on the website. So again, we're really lucky because with VIO, we have established a sort of protocol. We know where to refer them to for them to get some support that they need, because we as a sponsor, we cannot provide the drug to patients, and you know, we want to help them as much as possible, but it's not something that legally we can do. So now we have a process also established in place, and that is something that we can also use to give support to these families that reach out to us, because that is one of the consequences of having large dissemination in the public. So I would say that another advice is get ready for the consequences of whom you disseminate to and what you disseminate. We disseminate results from our clinical trial. We are excited about it. The news, they're trying to translate that into understandable language, but people have a real unmet need for better drugs. So be prepared for that. In our case, the VO really supported us, and people can write directly info@vo.net, and they can get support with open clinical trials, so they can get access to trials that may be more appropriate to their specific disease. So, okay. These are just recent communication and dissemination activities that I've done. I've mentioned before, there are some campaigns. One that I particularly liked was Conocelas, which is basically, it's from the ASECA. It's an organism, it's a Spanish research association for cancer that has taken this initiative to contact women scientists and ask them to present in schools, in high schools or primary schools. That's another dissemination activity. I mean, so I participated a couple of years ago. People from the lab participate every year. And it's a really fun way to disseminate our research, or part of it, or even just disseminate the fact that we are doing research and that people get to know us a bit more. With Peptomyc, we've had the chance to receive some other prizes. I've presented at some conferences. I think this is something that is accessible to all. And we also had the chance of being invited by the BioEurope Spring organization to present a sort of retrospective with Laura of our journey so far. So that was another activity we were just invited in. With the funding agency, this accelerator grant that we got last year, actually, we also could participate in other initiatives. They organized a sort of mission to go to the United States, learn about the market, and there were pitching sessions and training sessions there. So with the CFO, we went on a mission in California 1 month ago to train our pitching skills. And there we also had the chance to meet other people from the European industry and from the American industry. So that's another opportunity. With that, I would say what I'm trying to illustrate here is that we were not picky on how we disseminated. We just took every opportunity we could. We applied to grants. We see that they have programs. We try to apply for it. So I think it's really about investing time, be willing to invest time and energy. And that's not always easy to juggle with when you have a lot of priorities. But it's what enables you to really reach society and that people get to know you. One thing I wanted to mention is when we apply for these grants, there is often a lay abstract, and there's often now, it's quite popular, it's a impact statement. The question is often, how is your research going to impact society or policy, something like that. And it's often a question that is tough to answer, but I really encourage you to try hard to answer it. Sometimes I admit that last year I have even used ChatGPT to get some ideas on how to answer that question for my project. So trying to use tools to find key aspects that you can explore on how it can impact policy if it's not obvious to you. But it gave me some starting points, starting ideas on, in addition to impacting the life of patients, which is really key, and this is really the central point, our focus, which is clear, what else can it impact? So for instance, the fact that we collaborate with a lot of research institutes, with a lot of oncologists, we're strengthening the European network. Can this lead to impacting policy and society because of the way we use our drug in its clinical development plan? I mean, these are questions that are worthy of exploring, because in the end, people from these government, they don't just ask this question for asking it. They actually want to use the answer to see if there's a need to put efforts in some policy changes. Okay, I wanted to come back to this question that I've asked you before, and just use maybe the last 5 minutes or so to survey with you the challenges, the main challenges, and maybe some commonalities that we can discuss in consequence of that. Let's see here. Okay. So we've talked about terminology, knowledge gaps, and data overload. I think that for data overload, it's really about differentiating our project again, compared to the others. Communication channels, I've mentioned several, one, and you know them already, but so the communication agency from the research center, and then what everybody from the team can contribute. Limitations to access public. If that refers to publication, again, we talk about open access publications that can be important, or if it's about how to access journalists and newspaper and so on, I think that it's through the communication agencies, the best way. To get a good communication department, yes, couldn't agree more. Effective communication channels. Explain AI without scaring general public. I think for AI and for other topics, I mean, gene therapy is the same thing. So being aware of these ethical concerns, which are real, and how they can be addressed. And I think that is important, as we were mentioning before, discussing with our relatives, but also with experts to prepare the message appropriately. I think that could be a good advice.
And when communicating with, this is something you said, you had the capacity to connect the patients that were asking you about the project, the product, with people who can help them.
Yeah.
But at the start, I can imagine. I have been on that side also when I was uh, in other ventures. We had the same thing, no? When you are developing a drug or you are managing something, you get constantly asked, what about the start? But because it must be shocking at the start, the people start contacting you, they are really in the need. So, did you get any feedback from your communication department, or was you at the company created a modus operandi to know how to work, and that was it?
I mean, it was really generally people write to Laura and to myself because we have
Yeah,
our email address on everything and on the website. So that's what
yeah,
we find. And we always try to answer in the same day or minute or hour whenever it's possible. So
okay,
we try to answer fast. And it really depends. It depends on the patient what they ask. Some of them they are really desperate and they need support to find treatment. And in that case, the VO people, they are really the best one to help them. They are the medical oncologists who can review their medical dossier. We cannot do that.
no, no,
What
for
we
sure.
can do is answer their question when they have questions about the drug, development status, mode of action. I have this mutation. What does that mean? I mean, whatever is not medical advice, we try to do communication there too. I have, for instance, this patient from that contacted me on Facebook. She just wanted emotional support, and for her, it was just I was taking a couple of minutes for several days, weeks, months. Now it's been years. Her husband died of lung cancer, but it was just knowing that there's real people doing the research there made a difference for her. So sometimes it's just that it depends on the people. It really depends on the people. But yeah, the VU support, it's been invaluable for us, and it's been Laura and me aligning with them. This is the route that we should take. We should always recommend them to this and make sure that they get answer for their query as efficiently as possible.
Okay. And sorry, Marie, to cut you because I wanted to ask you also something else. I have an additional question because, as we have seen, the company has been working on bringing the drug to the market for several years now. Now that you have started phase two, right?
Mhm.
Maybe, yeah. Am I right? This is the last point I left the company. So when I was working with you, so in your opinion, I mean, now that you are having impact in patients' lives, um, do you think it was worth all the I mean, for sure it was, but all the suffering in business, not getting funding, not getting rejected. So what is your vision? So I think that's a lot of resiliency, bringing something to the market and something as a drug where many people fail.
Yeah.
What's your vision there?
I mean, there's still a lot of suffering. We still get rejected a lot. So yes, it's a lot of resiliency, but it's worth it. It's I mean, as long as we believe that it's worth advancing this drug to patient and it can make a difference in their life, we're gonna do everything we can to push it to the next level. So as you said, we started our second clinical trial. It's actually a phase 1B. We are testing it in combination with standard of care chemotherapy in pancreatic ductal adenocarcinoma patients. And we are at the beginning of this trial. We have started to treat patients and now we have to wait. And it's a bit of a torture because everybody is waiting. The pharma are waiting, we are waiting, the investors are waiting. So it's just, but we're doing everything we can in the meantime to see other combinations we can explore, preclinical data generation, other applications of the drug. second generation of the drug, variants, many different things that add value that in an effort to multiply our chances of success. But it's a high-risk business. I did not touch on that because I assume it's been covered in the other sessions, but 9 out of 10 projects fail. So 1 out of 10 may make it to marketing authorization, and then from marketing authorization to commercial success is another, I don't know what's the percentage of success there, but it's also relative thing. So it's a very risky business, but yeah, it's worth every tear, every awakened night for sure. Okay, we don't have much time left, but I think that we have covered many of the different aspects. I mean, that was just a list of what are the main challenges in communication and strategies. We've discussed some of them through the presentation, but I think that one aspect that I wanted to tell you is about effective storytelling. I hope that through the presentation, you've seen that there are some storyline, or there are examples that I've used, or images that stuck with you, things that we use that make it understandable or make the concept more relatable. At least if it wasn't a good example, find yours, but storytelling is key. Multimedia use whenever it's possible, videos, graphic, public engagement, we've talked about that. Transparent communications to improve trust of the public into the scientific results. So we cannot control what journalists say about our results, at least to a certain extent. Sometimes they let us read what they write before, often they don't. So you lose control, and you need to accept that this is the way things work. They do that for sensationalists, they do that to have a wider audience. You need, as a scientist, to be responsible enough to write with a certain transparency and not blaming anyone, but acknowledge that this is happening. Collaborations, trying to bridge the gap, education. So again, participating to campaigns for education, it's part of strategies that you can use. Then I had something about charisma, but yeah, I wanted to ask you if you think that charisma is innate or acquired. And but since we don't have time, I'm going directly here. You can develop charisma. There's this nice reference book that I recommend, which is different strategies to become a better communicator in general. So that's one great reference. And for storytelling, the work of Lisa Krohn is really the reference, I would say, to learn about storytelling. So these are some references I wanted to let you with. And then that's my last slide. So there's no such thing as a self-made man. There's always a way to build through teamwork and to learn. So it's that. If I have seen further than others, it's by standing up on the shoulders of giants. So we learn from the experience of others, and I encourage you to do the same for dissemination and communication. And that's it. I'll stop sharing the screen.
Thank you so much for these last recommendations, because they are really helpful. And yeah, I would like to thank you, Marie-Eve, for the session. I think it has been really interesting for the audience, because you have shared what has been the struggles, how you manage this through the way, to get by. And I think that's it's relatable to the health tech, because health tech, you were talking about your project, which is drug, but health tech is more or less the same. And resilience and good communications are key to make the project reach further. So from my side, I would like to thank you for your words and your time, and it has been amazing having you.
Thank you so much. Thank you for the invitation. And yeah, looking forward to meet with you again at the next occasion. Good luck, everyone, for communication. Don't hesitate to reach out if you need anything. If I can help with anything, I'll be happy to.
Thank you so much.
Thank you very much, Marie.
Have a great afternoon, everyone.
Bye.